Good morning. Thanks. One of the things that I'm going to be talking about is the lessons learned from the single analyte tests, and I think the short lesson from this is that I hope we don't have to do it much longer. I'm going to talk about three different topics. First is the EGFR mutation testing. This was the first one that we began doing in a CLIA environment starting in 2004, a decade ago. I want to talk about what happened when we introduced BRAF and HER2 testing, which there are also mutations that are commonly found in adenocarcinoma of the lung. And lastly, I want to talk about chromosomal rearrangements. Now, the lessons I'm going to be talking about is, one, doing testing in a CLIA environment, in an environment as opposed to one-off in your laboratories. And I want to talk about some of the logistics that are involved in having to go back to the same pathology specimens to do tests over and over again as they become clinically relevant. This starts in 2004 when there were 3 publications in 2004 linking mutations in the epidermal growth factor receptor to exceptional responses to gefitinib and erlotinib, which are 2 of the TKIs that Adrian talked about that began to change the landscape of lung cancer treatments. This was done because we began seeing dramatic responses that would go on for a year or longer, which was extremely unusual in lung cancer at the time. And it turns out it wasn't that complicated. They were TKIs directed against the epidermal growth factor receptor, so we sequenced the epidermal growth factor receptor and ended up finding those mutations, which were associated with rather dramatic responses. This prompted us in all of the institutions where this happened to set up our own EGFR mutation testing. I'm going to talk a little bit about that. This was done in research laboratories. It was not done in any kind of environment that has all the laboratory controls and such. The other thing that had happened was that on the science one, which was the one I was involved in, it was all done from paraffin-embedded blocks, which in 2004 was actually a little bit unusual to be able to sequence from. The one from Mass General, David Christiani, who was an epidemiologist, had collected frozen specimens since 1992. And for some of the people who responded, they had frozen tissue to be able to sequence from. And for the folks from Memorial, they ended up aligning themselves with Washington University to get their sequencing done. And it came out a little bit later. Now, one of the things that we ended up doing was after having performed this in a research laboratory, we had to transition it to a CLIA environment, one that is certified to do the laboratory testing. At the time, our pathology department didn't have the capabilities of carrying out sequencing on a routine basis. So we turned to the Laboratory of Molecular Medicine, which is the Harvard Medical School and Partners Healthcare Center for Genetics for the EGFR mutation testing. At the time, they were the group that was doing the germline testing for identifying familial syndromes of cancer and had the whole system set up. So it was a little bit of a unwieldy process where at the Brigham and Women's Hospital, which is our pathology department, they would take the slides, the pathologist would review it, they'd find 3 to 4 slides that had more than 50% tumor and send those unstained slides over to Cambridge, which was the laboratory. They only sent it twice a week because they couldn't afford to send couriers more often. And then they would do the testing, they would get a tracing, and then it would get sent back to the Brigham Path, and then it would be put into the medical record after the pathologist interpreted the signal, and it would show up. And this process took pretty close to a month. So the only patients who we were able to get information on is usually people who either weren't very sick, A, or B, people who had some site that needed to get irradiated for a few weeks and you had the time to do the testing. Luckily, since we're 2 different institutions, Dana-Farber, Brigham, and MGH, MGH pathology ended up not wanting to send their slides, so they made DNA and sent the DNA over there because they had pathologists at that Partners Healthcare Center. So that's how that worked. And it was very unwieldy. It took about 3 or 4 weeks, but it was done in an environment where you could put, and they had the custody so it would show up in the medical record. The license to do the EGFR mutation testing to link it to treatment, an exclusive license was granted to Genzyme Corporation. And in September 2005, they put out this press release. So it took about, the original announcement of the mutations was in April of 2004. And it took about 15 months between when that discovery was made before there was a commercially available test. and it was exclusively licensed to Genzyme Corporation, who then set up this test. And I went to the internet before this, and I looked to see what they had, and they talked about cells, and they described the process. And the other interesting thing I thought is that they said they had a team of 17 surgical pathologists and PhD geneticists to review the mutational analyses. Now, we spoke with them, and I knew the person who was in charge of setting this up, and they said they did less than 100 the first year they had this. So although we wholeheartedly embraced this and thought it was going to transform the care, probably appropriately, the rest of the oncologic community was somewhat skeptical, number one. Number two is, I assume Genzyme didn't make a ton of money as a diagnostic company having their team of 17 scientists to see this and review. And I suspect some of the people here who work for companies, this would probably sort of be a nightmare scenario. Now, the other thing we were doing is, as I mentioned, the criteria for this is it had to have greater than 50% tumor cells to be able to send this over, because the technology that allowed one to do this required a high proportion of tumor cells. And more than half of the patient's diagnostic specimens didn't meet the criteria. So it's not a very good test if you can only do it on half the patients. So we had something at Dana-Farber called the Translational Research Lab. So we set up something where you could detect the mutations in there. And I don't want to go in this too much detail, but what they ended up doing is being able to take the DNA from the tumor specimens and then ending up PCRing up the DNA and then annealing it to a wild type sequence. And then with the mismatch, they had a endonuclease that would digest the mismatches and then run it out on a column. And these dots, these little bumps here represent mutations in the sequence. And then you could take the DNA back out and then sequence it and come up with it. And this would detect mutations down where it was 1%, or at least less than, they could detect mutations less than 10% of if the tumor cells were the minority of it. Now, one of the things that happens is this was done in what we call the Translational Research Lab. It wasn't CLIA certified, and I'll mention what happened as time went by about doing these one-offs in your own laboratory as an indication for trying to do a sequence to guide therapy for your patients. Now, one of the things that happened is that this represents a waterfall plot. It's similar to what Adrian showed us. And one of the things that he showed is what the existing therapy was in 2000, in the year 2000, where we had less than a 10% response rate and a median time to progression of about 2 or 3 months. And this is a relatively recent one, but I wanted to show you this about how this transforms the care for this group of patients. Now, this is a waterfall plot. This represents no change. And this is a new EGFR TKI, and I'm going to talk about this at the end, about how this has been introduced. And you can see these red dots represents people with a partial response or a shrinkage by more than 50%. And our existing efforts for patients at the time was a response rate of about 20 to 30% in upfront and less than 10% in relapse patients. But the vast majority of these patients have regressions if you identify the mutations and then treat them with the epidermal growth factor receptor tyrosine kinase inhibitor, afatinib. And this was what was driving it. And we were of the belief, starting in 2004, that everybody who had this should start getting treated. The scientific community was somewhat skeptical until a series of prospective trials are done by 2009. Anyway, so there's this dramatic response. We thought it's important to be able to do this and to offer it to our patients in a consistent way. So the thing we learned about the EGFR mutations, they were discovered in 2004. We thought we needed a clinical test to be able to treat the patients, and we thought the rest of the scientific community should also have the opportunity to do this. It took about 6 months to get a test up and going, which is still about the length of time for us in our own pathology lab. It took about 1.5 years for it to get up and going in a commercial lab so people outside of our research institutions could do it. And almost very few in the oncologic community embraced this with the kind of enthusiasm that the academics who'd done this, and the testing really didn't start for another 4 years. I want to talk about BRAF and HER2 testing, and one of the things that typically happen as you transition from treating people up front, which is the standard had been chemotherapy, is when we first test these people, it's typically patients who'd had the initial standard therapy, and then we typically treat them in a relapse setting. The diagnostic specimens are usually initially there, and one of the things we embraced from the time we started doing this is that we thought it was important to begin testing as many genes as you thought were pertinent as soon as you had any hint of activity, so you could have the time to generate the information in a CLIA environment and know that it would be useful, because it's much easier to do this as one initial test with multiple tests rather than doing these one off at a time as you think they might be needed. And BRAF and HER2 are the next set of testing that we did, and I'll show you why. Now, starting in 2007 and 2008, we began knowing that if you had agents directed against the specific mutation of V600E, which is the mutation that's present in about 80% of melanomas, if you treat them with a drug called Vemurafenib, which is specific for the mutant form of BRAF, you have a much more dramatic level of evidence of anti-tumor activity in these melanomas. Now, this began being observed in about 2007, 2008, and this phase 2 wasn't published until 2012. Once again, you see this waterfall plot, and as you can see, nearly all of these patients with melanoma have a regression when you use a drug directed against this specific mutant form, and it's not active if you don't have this particular mutation. And as you can see, it's quite dramatic. And it turns out about 1% of lung cancer patients have a V600E. So it's a little bit like looking for a needle in a haystack, but in those patients, it's pretty meaningful. EGFR is present in about 15%, so this is a 1%er. The second thing that we ended up seeing starting in 2008 is there were insertion mutations of HER2 that were identified in lung cancer that are present in about 2% of the patients. They're insertion mutations, and there were drugs directed against HER2. The first was neratinib, and there was a phase 1 trial combining neratinib, which is directed against HER2 and is used in breast cancer, and temsirolimus, which is an mTOR inhibitor. It's an agent directed against a downstream target of the activation form of the tyrosine kinase inhibitors. And this is a phase 1 trial, so it had multiple different types of cancers. And you can see in yellow are these non-small cell lung carcinomas. And of the 6 patients who were treated, 2 of them had responses. And this shows the level of activity in the HER2 mutants. And they had partial responses here in these HER2 exon 20 insertions. So in lung cancer and relapse setting, 2 out of 5 is not too bad, and the other 2 had stable disease. So we began thinking, starting in 2008 and 2009, it was important to identify both BRAF and HER2 because it had been shown in other tumor systems, and we had some early evidence of going after HER2 using neratinib, an agent directed against HER2, as well as the mTOR inhibitor. So what we ended up doing, because we thought this might be useful as soon as we found about this, we added BRAF, HER2, PIK3CA. I'm not going to talk about PIK3CA, but at the time we thought it might be active in breast cancer. And then at this time, and I'm going to come back to this, ALK rearrangement testing was added. So we also added KRAS because it was potentially a predictive marker for the absence of a response to the TKI. So we're up to 6 different mutations. And the other thing we did is that those who had been sequenced for EGFR and KRAS and were negative, we had to go back and retrieve all those slides or all the DNA and rerun all these tissues because it was a way of enriching for people, because you typically only have one of these, and we now call them driver oncogenes. So we added this in 2008. Once again, we used the Laboratory of Molecular Medicine because it was still our CLIA certified environment. And we ended up having to go back and either re-pull all those path specimens that had tested as negative or pull the DNA. Now, at this time, and we'll talk a little bit about this, is that we started something called the Lung Cancer Mutation Consortium. We added some additional ones, and we added MET and we added ALK, and I'll talk about these two, about the ALK and ROS1 chromosomal rearrangements. For the mutation testing, you could go back and either pull the DNA or you could pull the slides, and it wasn't, although it was difficult to find them, identify them, rerun them, in order to do the testing for the rearrangements, at the time we needed a FISH test, fluorescence in situ hybridization, where you require at least three 3 slides from pathology for each test. And the reason why we ended up doing this is something that Adrian showed, and that is this dramatic evidence of response to the ALK-positive patients. And secondly, one that came out this year is ROS1, which is another chromosomal rearrangement that was detected and described. And the patients began enrolling on this cohort in 2010. Now, interestingly enough, this is present at 1% or less. In order to get these 50 patients, it took 3 years from the two major medical centers to be able to come up with 50 patients to test these because it's present at 1%. Now, lung cancer, there's 240,000 people a year that get it. So even at 1%, you've got more than a couple thousand patients in the US a year who have it. Now, they end up with this FISH testing, and this goes into what they do and do not describe as being positive. This is the approved test. So the approval that took place in 2011 for ALK was also with a specific test, and it has to do with colors, and it has to do with how far, and they end up saying it's 15%, 15 out of 100 when they count these, when they see a separation of the 2 different colors on this fluorescence in situ hybridization where you look at the cancer cells. Now, it turns out it's not 15% because 15% of the cells are seen. It turns out we think all the cells have the rearrangement. It just has to do with the spatial relationship and when you take a look at these cancer cells to see them come apart. And this was really the first one where they approved both a drug as well as a specific test, and they specified that it was by the company and the criteria that was in there about how this was approved. Now, the same thing is for the ROS1. This is how it was identified. This drug is not yet approved despite that response rate. It has, to the best of my knowledge, this hasn't been submitted for approval for those 50 patients. Now, when we take a look at these landmarks, the crizotinib was approved in 2011. The ROS testing started in 2010. And so now we're left for routine of doing it is 3 FISH tests, which require a total of 9 slides. And you need a total of somewhere between 3 and 5 slides for the DNA tests, which in a metastatic setting where you have diagnostic specimens, you may or may not have those. And you also need a high proportion of tumor cells. So this starts, there's 2 nightmare parts about this. Number one is the amount of material you need for people with advanced disease to do this testing, number one. And number 2 is having to go back and retest these specimens that have been negative for everything else to end up finding these relatively rare subsets of patients. So this was where we were in 2011. And I'm going to go back to the EGFR mutation testing because despite sharing with you that we were all excited about this in 2004 and thought everybody needed to be tested, here in the United States, this actually wasn't approved. There was no drug approved for EGFR mutants until last year, 2013. And to me, it either speaks to our regulatory process or the ability of our docs to carry out appropriate trials to lead to registration. I want to go back to this EGFR with afatinib, which is one of the 2 drugs that are approved for EGFR mutants in 2013. And the interesting dilemma we find is that there's 2 drugs that are registered now, erlotinib plus afatinib for EGFR mutants. And interestingly, the erlotinib is approved for a COBAS test, which happens to be owned by Roche Diagnostics, which is the same company that makes, same group of companies that make erlotinib. And afatinib is approved for using a Quiagen test. So what you're left with, if you're actually going to be held to what the label says the drug is indicated for, and you don't know ahead of time which drug you think you're going to use, you would theoretically need to run 2 different tests because there's 2 different tests approved for 2 different drugs at the current time for the mutation testing. And the 2 different drugs, one is a COBAS test. These are both PCR-based tests. And the other interesting thing is if you do have a COBAS test and you get erlotinib first, the most common mechanism of resistance, and there's drugs coming on here, is that the most common form of resistance is a gatekeeper mutation called a T790M, and COBAS is not licensed to pick that up. the TheraScreen test is pick that up. So depending on the circumstance in which you need to do your testing to qualify people for a trial, you may need a CABAS test, or in the relapse setting, you may need a TheraScreen test. So those are our two different tests for the EGFR mutant. And here comes the Foundation Medicine test. This is the most common multiplex test being used. On the website, it says it costs $5,800, where it does 315 genes, and it says it can detect alterations, base substitutions, copy number alterations. So you can get all those tests in one fell swoop. And our folks here ended up starting in 2013 for doing what we call OncoPanel, which a lot of the different academic centers have, where you get all 300, where we get about 350 at one time. And I put in dark the ones that are pertinent to the ones that I've talked about, because it ends up generating information on all of these, which is ultimately where we hope to go. You order one test, and you'll be able to use it during the patient's lifetime. So I've gone through this timeline and the things we've learned from this. Having gone through sort of the nightmare of retesting everybody each time you think you have a new indication, to some extent it's a good problem, because there's lots of different things for which we have targeted therapies in lung cancer. However, the process of going through, and the thing to do is to try to test for every gene you think you might know during the patient's lifetime in one fell swoop. So thank you.
Thanks, Bruce. So we're going to continue the same line, which is 20 minutes each speaker, and then at the end we'll have 30 minutes for discussion. So we'll keep the questions to the end. We'll move from the single analyte to multivariate analytes or multiplex, and Mia Levy from Vanderbilt will talk about next-gen sequencing.